Let's start with the introduction. Variation in biology is any differences between cells, individual organisms, or groups of organisms of any species caused either by genetic differences or by the effect of environmental factors on the expression of the potential genetics. Variation may be shown in physical appearance, metabolism, fertility, mode of reproduction, and other obvious of measurable characters. Genotypic variation are caused by differences in number of structures of chromosomes or by the difference in the genes carried by the chromosomes.
Polyploidy meaning as an organism containing more than two pair homologous sets of chromosomes. There are euploids and aneuploids. It can occur spontaneously and common in nature for adaptation and speciation. Cases of polyploidy are: first, accidents during meiosis called as autopolyploidy results in the wrong number of sets of chromosomes in the gametes called as unreduced gametes. Second, contribution of two different species to a polyploidy hybrid called as allopolyploidy. It because non-homologous chromosomes cannot align during meiosis. Lastly, the chemical colchicine drug induced polyploidy. Colchicine causes meiosis produce diploid gametes instead of the normal haploids. This slide shows naming of polyploidy. Polyploidy have their own names, which is for each names of sex represent for the number of sets of chromosomes in the nucleus of a biological cell. Okay, first for one set named as haploid or monoploid, two set named as diploid, then three set and above known as a polyploid, which is three set named as triploid, four set named as tetraploid, five set named as pentaploid, six set named as hexaploid, seven set named as heptaploid or septaploid, then lastly 8 set for octoploid. There are also advantages of polyploidy. Polyploidy plays a significant role in plant breeding and horticulture. First, polyploidy comprise more strong influence than the diploids and results in the production of large-sized flowers and fruits. Okay, therefore, it has economical significance. Second, polyploidy plays important role in the evolution of new species. Third, polyploidy results in the changes in the season of fruiting and flowering. Fourth, polyploidy leads to the formation of new varieties that depict high resistance to disease and increase in yield. Fifth, teraploid cabbage and tomatoes consist of more ascorbic acid, whereas tetraploid consists of more vitamins A. Then lastly, both euploidy and aneuploidy in men cause congenital diseases. There are disadvantages of polyploidy as well. First is changes in cellular architecture and regulatory implications. Second is difficulties in mitosis. Third is difficulties in meiosis. Fourth is regulatory changes in gene expression. And lastly is epigenetic instability, which are divided into two parts, autopolyploids and allopolyploids.
Polyploidy have two types, which is euploidy and aneuploidy. Euploidy is an entire set of chromosomes is duplicated once or several times. Aneuploidy, the abnormal condition, one or more chromosomes of a normal set of chromosomes are missing or present in more than usual number of copies. This is different set of euploidy and aneuploidy. In euploidy, constitute the majority of polyploid. Then a complete set of chromosomes, the basic number is X. Then occasionally in animal and frequently in plant. In aneuploidy, particular chromosome reaching a set in abnormal condition, which is single extra chromosome, which 47 is trisomy. Missing chromosome, which 45 is monosomy. Result is egg or sperm having extra or a missing chromosomes, typically not inherited, and most trisomy occur due to the non-dejection. Alright. Type of euploidy have 4, which is neolisomy, trisomy, monosomy, and tetrasomy. All of them have their own number of chromosomes. Despite is about non-dejection and the origin of aneuploid. Non-dejection is failure of chromosome to separate, leading to unequal distribution of chromosome to opposite poles. Then the origin of aneuploid, abnormal dejection may occur at first meiotic division. Then at second meiotic division, producing a gamete which gain or lost in chromosome.
Haploid refers to those plants which possess a gametophytic number of chromosomes in their sporophyte. If successfully cultured by using anti-culture, the plantlets resulting will have a haploid genotype. Haploid genotype may arise directly from embryos or indirectly via calyx. To have maximum genetic variability in the plantlets, breeders usually use anthers from F1 or F2 plants. The plantlets are diploidized to produce diploid plants by using colchicine for chromosome doubling. Haploid may be produced by one or several methods, which is anther culture to induce androgenesis, ovary culture to induce gynogenesis, and embryo rescue from white crosses. There are few applications of haploids. The first one is development of new cultivar. Through diploidization, haploids are used to generate instant homozygous through breeding lines. It takes 2 seasons to obtain double haploid plants or 7 crop seasons using conventional procedures to attain near homozygous line. Heritability is high because dominance is eliminated. Next is selection of mutants. Androgenic haploids have been used for selecting especially recessive mutants. In species such as tobacco, mutants resistant to methionine analog of the toxin produced by Pseudomonas tabacci have been selected. Another application of haploid is development of supermales in asparagus. Haploids of asparagus officinalis may be diploidized to produce homozygous males or females.
So here is about androgenesis and genogenesis. Androgenesis defined as the process of embryo development from the male gametophytes, for example, microspores or anthers, with the subsequent regeneration of haploid plants from these cells. Meanwhile, genogenesis is the culture of unfertilized female gametophytes, for example, ovules and ovaries. This method used when plants do not respond to androgenic methods. There is a problem with regenerating albino plants from anther culture, or the donor plants are male sterile. Genogenesis is usually less efficient than androgenesis. It used in plant families that do not respond to androgenesis like Liliaceae and Compositae. This is the process on how androgenesis and genogenesis develop. Androgenesis. First is parent plant selection. Second is selection of flower bud. Third is microspore development stage determination, such as depistaining, stiff region, and acetocarmine or acetorsine. Fourth, surface stabilization of young flower buds. Then isolation of anther or microspore from buds. Next step is inoculation of anther on appropriate medium, and last step is regeneration and recovery of plant. Meanwhile, genogenesis. First, selection of healthy parent plant. Second, check ovary or ovule developmental stage by histology. Third, select appropriate flower bud and surface sterilize. Fourth, inoculation of ovary or ovule in suitable medium. Then incubation of ovary or ovule culture. Next is regeneration of calyx or embryo, and lastly is haploid plant recovery. That's all from us about this topic. Thank you for watching.